Welcome back to OmicsLogic Data Science. In this session, we will introduce biomedical data analysis techniques and approaches that you can master and adopt to help you conduct better research. The goal of this program is to introduce the need for data science and application to biomedical projects. Many of such projects require a good understanding of various omics data types, steps to process raw data and structure it for analysis, as well as know how to extract meaningful information from such data sets through visualization, analysis, and machine learning. Importantly, here you will learn to link biological phenomena with data patterns for meaningful interpretation. Before we go on, let's review the program plan and objectives. The syllabus of the program is organized into three different parts: introduction, methods, and applications. In the introduction, you will be prepared to deal with relevant terminology and key concepts to refresh your memory and get you started. The methods part will give you a practical sense of commonly used methods and functions for analysis of omics data, starting from exploratory to statistical testing, supervised and unsupervised machine learning, as well as deep learning. Finally, the project section will offer a sense of application for how these methods are applied to real datasets. Completing the full program will give you the confidence that you understand the analysis and can apply it independently to a dataset of your choice. Each session is designed to cover the topics in a practical and informative way. Oftentimes, these session videos are going to be sent to you by email. In each session, you will also find additional links to asynchronous resources that review and enrich the covered topic. Depending on how far you want to go with the program, you can choose to stay within the scope of the syllabus or explore beyond what is covered in each session with asynchronous materials that are included. In this session, I will try to cover everything you need to know to make the most of your time and benefit from this program. In the future, we will also schedule a Q&A session where you will be able to submit your questions and get answers in an interactive session. Before you jump into the data science curriculum, we do recommend that you complete the introductory courses which you also have access to, including the Introduction to Bioinformatics, Bytes and Molecules, as well as a basic overview of R and Python coding for bioinformatics analysis. If you're already familiar with these topics, let's discuss what specifically will be covered in each part of this program. If a lot of what we are talking about when we say words like transcriptomics, genomics, metagenomics does not really make sense yet, if you're not sure what omics data looks like, and maybe you have no clue what dimensionality reduction really means, the first part of the program will make sure that you're familiar with these topics and understand the terminology. We will try to answer questions about processing or preparing data for analysis. We will walk through the concepts behind finding signal in noisy data and explain why it is important to understand how the molecular characteristics of data objects can be associated with a concept that we can interpret. Here you will find three main options. There's going to be a logical approach, which will be based on analysis that will explain most concepts using Excel and the user-friendly T-BioInfo platform. You will have another track, which is called BioML in R, for those who are more familiar and comfortable with R and RStudio. And finally, you will see the BioML in Python, which focuses on the Python programming language. Each one of these courses covers similar topics, however, the level of complexity and challenges with syntax will be very different. We recommend that you take the BioMedical course first and then decide to finish either the Python or the R coding courses separately because the syntax is quite different from each other and it can become confusing if you start working on both at the same time. Coding exercises and syntax in each one of the coding lessons will rely on packages that have to be installed on your computer to run. However, we will also provide you with an online environment where you can read and understand the syntax, run the code on example datasets, and gain access to Jupyter notebooks to develop code of your own and submit assignments. Ultimately, you might choose to copy all of the code and save on your own personal machine. To make sure that everything is working, make sure that you're not assuming that code will work exactly the same way. There might be many different technical challenges. For example, you might need to install Python or R. Maybe you will need to set up your coding environment, which includes setting the working directory, loading packages, and make sure that you overcome the technical limitations which often come up and are specific to your own computer. So the safest way is to utilize the online environment that we provide, and after you are ready to transition and you understand how the code works, to try and set up everything on your computer. After completing this introduction, we will jump into machine learning topics, unsupervised and supervised learning, as well as feature selection. These methods are often used for various tasks, such as visualization, organization of data, finding signal in noisy omics data, as well as things like predictive analysis and feature selection. To be able to use these methods, you need to understand how they work. What are their inputs, variables, settings, and available packages that combine multiple functions to make the methods easy to deploy. You will learn how to load data, select a method, and run it to get an output of that particular model and try to understand what it found. Then we will start our review of additional analytical methods that are useful for biomedical discovery, including network analysis, such as pathway analysis, generalized linear models that are used to distinguish between expression profiles of transcripts, as well as deep learning models that can be used for specific types of situations to improve accuracy and decrease feature engineering that a lot of conventional machine learning relies on. We will also talk about the need to combine various machine learning models into a series of steps to achieve more sophisticated tasks. And finally, we will talk about different use cases that can show how these methods that we review have been used by researchers to find meaningful insights in complex experiments, biological samples, animal models, or even patient data. With the few examples that we review as case studies, you can then develop a project of your own or follow asynchronous tutorials to reproduce an analysis that you can improve upon, turning it into a novel and personal study. On the OmicsLogic portal, you will find a more detailed review of each dataset that we use in our exercises with additional tutorials, method clarifications, and links to research articles where the authors demonstrate the utility of machine learning methods for their own findings. Now let's take a brief moment to explain what kind of resources you will have access to during the program and how exactly to use them. One of the major challenges in omics data analysis is related to accurate, reproducible, and effective processing of large-scale datasets that represent gigabytes or even terabytes of data. Such large quantities prevent many people to gain practical hands-on feel for these pipelines, which are critical to understand where the data you analyze comes from. To be able to do that, we have prepared a number of example projects that are essentially demos. These pipelines do not have to run, they don't use computational resources, and they demonstrate the inputs and outputs, as well as the logic of analysis for each example. You will also find that a lot of these pipelines are color-coded and provide a very clear explanation of each step with conditions, methods, and settings that you have to know for reproducible workflows. You will also get to practice all of this code in the online console, where you will find predefined answers and you can try to run the code simply by modifying it according to what was explained in the syntax section. You will find a lot of these clear explanations in our courses. For example, this course on Introduction to Data Science in Python and Introduction to Data Science in R. You will find that many of the same methods are covered, but with little variation in the way the data is prepared, analyzed, and the outputs that these methods produce.